Hi guys, just taking you through the Robert May self-study guide. So let's get right to it. Here's the table of contents like you're probably used to by now. You can see this one isn't quite as bad, including your study questions. It's only about 44 questions. This one, at least from my perspective, focuses a lot more on immunology than it does on microbiology. So hopefully that's something you guys are interested in. Okay. So what do we have here? So let's start with our introduction to Robert May. I'm switching the game up on you guys a little bit. I'm not giving you the paper case this time. Instead, you have about a 2.5 minute video where Robert May comes and tells you why he came in today. Watch the video a couple times. It's only 2 minutes long, so that shouldn't take you very long. And take some notes about what he's saying, what he says about his history, how he's feeling today, his anxieties, any behaviors he notes. Make a list of that and be ready to discuss it when you come to class. When you come to class on, I think this one is taking place on a Thursday. So when you come to class that day, you'll take your readiness assessment. And then right after the readiness assessment, you'll be able to download a paper case from Entrada. We're going to give you the entirety of the case from the time the patient shows up through any lab results, x-rays, diagnostic tests that are given, through kind of the diagnosis of the microorganism and the diagnosis of his disease. So you're going to get all of that at that time, and you can go through and review it. And then we're going to utilize it during the session to kind of dig into parts of it a little more deeply. Okay. All right. So that's the case. We're going to start with micro. We're going to talk about organisms related to pneumonia because Robert May shows symptoms of pneumonia in his video. As you'll be able to see, he's had pneumonia a couple times before, and he's pretty scared of it. There are some organisms that are associated with pneumonia, some of which we're going to cover here, some of which we won't. You're familiar with the streptococci, but you haven't learned about all of them, and strep pneumo is not one we've gone into great detail on. We will in a couple of cases, but that's kind of the most common cause of pneumonia in the United States. There are other major causes of pneumonia like Haemophilus influenza, Legionella pneumophila, Staphylococcus aureus, Chlamydophila pneumoniae, and Mycoplasma pneumoniae. All of these can also cause pneumonia. Klebsiella pneumoniae we're actually going to talk about in this self-study guide along with Legionella pneumophila. Klebsiella pneumoniae is one of the ones that we associate specifically with patients who suffer from alcoholism and patients with a history of type 1 diabetes. Okay, so we go through a whole bunch of random bugs here. There's the Bartonellas, which cause some things that are similar to pneumonia and some that aren't. So like B. cantana, B. hensley, and B. bacilliformis. You can go through all of those. They're pretty rare. There's brucella. This is one we actually associate with people who spend a lot of time with livestock. And then there's Legionella pneumophila, which I go into pretty good detail on. There's a video for it here. You can read through all of this, but there's 2 different types of diseases. There's also a sketchy micro video for Legionella, which I recommend. It's pretty good. All right, then we're going to talk about Klebsiella. This is the first of the Enterobacteriaceae. The Enterobacteriaceae are a really large family of bacterial organisms, and we're going to talk about the rest of them in the Jenna Wilson case, but we're going to start talking about them today in the Robert May case with Klebsiella. So we talk about some lesser known versions of Klebsiella as well, like Oxytoca, and then we also talk about K. granulomatis, so you can go through those. And I just put in a little plug for Streptococcus pneumoniae and for Staphylococcus aureus, because both of those are causes of pneumonia. Then we go into some viral causes of pneumonia. Again, we're not covering the most common causes of pneumonia from a virus right now. One of them is actually influenza, which you guys have already covered a bit and we'll cover again in the future. So we go through the Bunya virus family, Hantan or Cynombre, and then we move into immunology. So from a micro standpoint, this case is pretty small and your main focuses are Klebsiella and Legionella. Okay, so immunology. We're going to move into the adaptive immune response. So we've been spending time on the innate immune response, kind of those initial events in the immune response that begin to kill off the pathogen, but also eventually will lead to the activation of the adaptive immune response, which is very antigen specific and has memory. So how do we move into that? And remember, the adaptive immune response is entirely populated by T and B cells. So you get another kind of introduction to the difference between humoral and cell-mediated immune responses. Excuse me. This is where we're actually going to dig into that. So you see there's a lot more details here. Then we go into antigen presentation. The concept of antigen presentation is a complex, elaborate, fascinating dance between two cell types that I could pretty much watch forever. But since you guys might not share my enthusiasm on that, I've tried to break it down as simply as I can. But basically how the cells move throughout the body using lymph nodes, I go into that here. So the lymphocyte recirculation, both entering and leaving the lymph node. And then we have to talk about the antigens themselves. So we've discussed the term antigen a lot of times, but now we're going to go into what it means to be an antigen. How does an antigen lead to an immune response? What part of the antigen do the cells see? Things of that nature. Okay, now we have to talk about something really important. We need to talk about MHC molecules, major histocompatibility molecules. These things kind of run the immune world, and they also wreak havoc for transplant surgeons. They were founded because when we tried to transplant patients, they would get rejected. And one of the main reasons was these MHC molecules. And if you don't match these up, then you don't have a successful transplantation. But that's not actually what they were designed for. They were designed to serve as the silver platter that serves antigen to T cells, because T cells can't see their antigen outside of MHC. You need to know your MHC. I've made a video here for MHC. Watch it once, watch it twice, watch it a couple times during this block. These are an important molecule, and you need to understand how they work. You need to know their names. So for MHC class 1s, A, B, and C, so one letter. Where they're found for MHC, that's all nucleated cells, and what they do. MHC class 1 presents to a type of T cell known as CD8 positive T cells. These are killer T cells, and it does that using antigens it finds inside the cell. Why? Because sometimes we have tumor antigens or viral infections or things like listeria that hang out inside the cell, and we need to see those antigens and kill cells that are infected too. MHC class 2, there are two letters for MHC class 2, DP, DQ, DR. So that's an easy way to remember it. You're only going to see these on professional antigen processing cells, and then they present to CD4 positive T cells, and they present exogenous antigens, so like bacterial infections found outside the cell, like Klebsiella or Streptococcus pyogenes. These are Strep pyogenes is an organism that doesn't go intracellular. So go through that. Here's a table. Then we go into T cell receptors. So I've told you that T cells are antigen specific and that they might have millions of T cell receptors on their surface, but they're all specific for the same thing. So go through this. It's going to take you a little time. Pay attention to it. Look at this structure. Understand the components of this structure. Contact me if you need help. This stuff is complex. Now we're also going to get into lymphocyte development here. This session and next session, we're going to talk about how we make T cells and B cells. And a lot of that is tied to how they make their antigen receptors and how we keep those antigen receptors unique. This is difficult to understand, so contact me if you have issues. This one's tough, but you need to know the stages of the T cell development and how it kind of moves through. So I've outlined them here. And then you also need to know the components that make up the T cell receptor. So I don't go into that too much here. We'll go into it a lot more next time, but that's the V, D, and J regions. Okay, in this video alone, I've said CD4, CD8, CD3. I've said a lot of these CD markers. Kind of like cytokines, CD molecules there's just so many of them. I think there's something like 112 at this point, probably more. So I go through them in a video. This is another one of those videos like the MHC video and the cytokine video that you're going to want to watch a couple of times throughout the block, and it'll make more and more sense the further we get into the immune response. So give it a listen now. It's okay if you don't know all of the CD molecules I'm talking about right now. For now, if you know what CD3, CD4, and CD8 are, I'm pretty darn pleased, okay? Later, we'll worry about the rest of them. Oh, and CD14, because we've talked about that. It binds LPS. It's pretty important. Okay, moving on to nutrition. Dr. Peterson has created a self-study here about adult malnutrition. So go through this, what you might see as the role between nutrition and the immune response and the type of nutrients that could be lacking in a patient like Robert May. Dr. Poole has you going through mechanisms of cell injury and death. He's created 3 fairly short videos for you here. He also wants you to, oh, he put the times in here for you. He also wants you to just kind of go through the reading on pathways and mechanisms of cellular adaptations, and you can find that in your Robbins Basic Pathology 10th edition. I will make sure to hyperlink that before I post this document. Okay, pharmacology, you are going to learn about tetracyclines and fluoroquinolones. These are 2 different types of antibiotics. I believe they are largely involved in inhibiting either protein synthesis or nucleic acid synthesis. So make sure you understand how these work. Dr. Bradrick took this mostly from a textbook, so contact him if you have any further questions about that. One of the things to keep in mind when looking at these is knowing which drugs are in which group, and then also what the toxicities and side effects of any of these drugs are, and if there are any interactions with food or how a patient might respond to these drugs. Those are things you might want to pay attention to. Okay, and then the last, I think it's the last discipline in here is psychiatry. Dr. Dance wants you to review alcoholism. So Robert May, our patient, has some history of alcoholism that you'll learn more about during the case. So we want to go through how alcohol affects the body and the neurologic complications. And as I mentioned, K pneumoniae is associated with pneumonia in alcoholics. So he does provide an article link here. You won't need this article link necessarily for your readiness assessment, but you will use it during the CBCL session. So make sure you're just aware of where to find it. But the rest of these you should keep an eye on. Particularly for this session, make sure you're looking at your study questions. Use your study questions to guide your reading of these articles because they're rather long and you don't need all of the details from them, but you do need to be able to pick out the information that's listed in those mastery questions, okay? And here are your mastery or study questions. There are 45 of them that take you through the whole document. So 1 a page, something like that. And here are all of the resources that we used to put this together. Once again, you can find the discipline directors and their office hours in Entrada, and let me know if you have any further questions. Good luck.